welcome to today's episode. As you probably know, molecules like proteins aren't stiff or static. They bend, twist, and breathe. This flexibility is not just a detail, it is fundamental. Proteins need to move to do their jobs, whether that is jump-starting chemical reactions, passing messages in cells, or helping us fight off disease. But here's the catch. Predicting that flexibility quickly and accurately is not easy. That is why I am genuinely excited about today's topic, a brand new tool called Pegasus. Pegasus is designed to do something quite interesting. It can predict how flexible a protein is, just from its amino acid sequence. No 3D structure, no simulation, just the sequence. We are going to unpack the science behind Pegasus by diving into the article by Yann Vander Meersche and colleagues published in Protein Science in June 2025. Traditionally, if you want to measure protein flexibility, you turn to biophysical methods like X-ray crystallography or NMR spectroscopy. These are very powerful and they've taught us a lot, but they come with some drawbacks. They are expensive and time-consuming, and while they usually agree on where a protein is flexible, the actual numbers they produce don't line up easily. X-ray and NMR capture different kinds of motion on different time scales, and that makes their data tough to merge and use for statistical modeling. Plus, flexibility measurements can also vary depending on the experimental setup. So here is the situation. We've got millions of protein sequences, but not much motion data. That is where molecular dynamics, or MD simulations, come in. These simulations show you how every atom in a protein moves over time, giving a more uniform picture of flexibility. Now, MD simulations are powerful, but they're also computationally expensive and time-consuming to run, especially if you want to cover thousands of proteins. So here is one clever part among many. The team behind Pegasus used a collection of MD simulations present in the Atlas database as training data. Atlas is a curated collection of high-quality MD simulations of hundreds of globular proteins. From these, the researchers extracted flexibility measurements and used those as ground truth labels to train Pegasus in a supervised learning setup. Using that training data, along with cutting-edge protein language models, or PLMs, and deep learning, Pegasus can predict key measures of flexibility directly from a protein sequence. One of those measures is root mean square fluctuation, or RMSF. In this package, it means how much each alpha carbon, one of the backbone atom in every amino acid, tends to wiggle around from its average position over time. But let's go back to protein language models, or PLMs. What are they, and why are they such a big deal in Pegasus? PLMs are deep learning models trained on millions of protein sequences. Basically, they learn the language of proteins, picking up on the patterns and rules that occur naturally in biological sequences. Once trained, these models can turn any protein sequence into what is called an embedding, a long string of numbers, maybe 1,000 or more, depending on the model, that represents each amino acid, in a way that captures a ton of hidden information. Things like structural tendencies, functional regions, and even long-range interactions in the protein chain. Older models often relied on simple statistics, but these PLM embeddings are rich and context-aware. They pick up subtle signals that traditional methods can miss. And this is where Pegasus gets really smart. It takes the embeddings from these PLMs, which are frozen, meaning they are not changed or retrained, and feeds them into a separate deep learning model, a neural network that's trained to predict flexibility, thus values like RMSF and other motion descriptors. It learns to map those embeddings to real motion values using the simulation data from the Atlas database as ground truth. What's even cooler is that Pegasus doesn't rely on just one protein language model. It uses several, like ProtT5 and evolutionary scale modeling, or ESM. Each one gives a slightly different perspective on the protein, so Pegasus trains a model on each of them and then averages their predictions to get a more reliable final result. Let's talk about performance. It is really impressive compared to, say, a previous tool called PrediFlexi. Pegasus shows a big jump in accuracy for RMSF prediction. We're talking like a 24% gain in Pearson correlation. And what's maybe even more striking is that it outperforms another tool named Medusa, even though Pegasus was trained on a data set that was 7 times smaller. It really shows the power of using those PLM embeddings here. They seem to capture so much information. So, okay, impressive performance, but beyond the numbers, how useful is this for researchers? Well, its utility is pretty broad. For instance, its predictions correlate well with experimental measurements of protein disorder. Its predictions correlate also with AlphaFold 2's confidence scores. But Pegasus offers sort of distinct dynamic insights. And crucially, it's much faster. It is a very lightweight alternative if you need to estimate flexibility of a protein quickly. And also think about large-scale studies. That speed difference is huge for practical applications. And speaking of applications, the paper shows also some really cool real-world examples, like with point mutations. The authors demonstrates how Pegasus can predict flexibility changes when you have even just a single amino acid change. So Pegasus can help understand disease and help engineer better proteins. And you can try this out yourself. There is a free web server. You can put in a protein sequence and get predictions almost instantly. You can even submit batches up to 100 sequences. And for more advanced users, there's a standalone version too. So wrapping this up, what's the big takeaway here? Well, Pegasus really feels like a powerful new way to get insights into your favorite proteins, especially when you want to understand how they move and flex without running long and complex simulations. Understanding how proteins move is fundamental to understanding health, disease, and how potential drugs might work. So here's something to think about. Imagine using a tool like Pegasus that predicts these intricate protein dance moves just from the genetic blueprint. What if we could use that predictive power to actually design completely new proteins, proteins with specific custom-made flexibility for new medicines or targeted industrial jobs? The potential for protein engineering is definitely exciting. That's it for today. Thanks for tuning in. Until next time, take care.